Bonjour à toutes et à tous. Donc, je suis vraiment ravi de vous retrouver pour la troisième session du colloque international Recherche fondamentale sur les cancers de l'enfant. Donc, je vais animer cette session. Donc, je suis Olivier, je suis chercheur au CNRS et à l'Institut Curie. Et je vais co-animer cette session avec Pierre-Yves DEMOURES, qui est représentant des parents et patients du collectif Gravir. Donc, Pierre-Yves a longtemps travaillé avec Patricia Blanc à Imagine for Margot et est toujours un bénévole très actif avec Patricia au sein de cette association. Donc Pierre-Yves, je vous vois. Bonjour Pierre-Yves.
Bonjour à tous. Bonjour Olivier.
Parfait, donc tout va bien. On vous a en vidéo et je vous vois en visu. Donc je vais maintenant passer en anglais pour démarrer la session scientifique. So for this third session, we will address one really important question related to big data. So we have heard yesterday and today about new technology, sequencing, bioinformatics, and so on. And in this session, the goal is really to address specific questions such as what is big data, what for, and what will be the future. So to help us to understand and to explore big data, we will first welcome four prestigious speakers. Each of them are really leading people in their respective area, and I think we are really lucky to have them today, and I really want to thank them again. So first we will welcome Dr. Stephen CHANOCK, Director of the Division of Cancer Epidemiology and Genetics at the National Cancer Institute in the US. So Stephen is a world-leading expert in the discovery and characterization of cancer susceptibility regions in the human genome. He has just an incredible list of publications and received numerous awards for his scientific contribution to the understanding of common inherited genetic variation associated with cancer risk and outcomes. So thank you, Stephen, and you can start if you can. Everything is okay?
C'est bien. Merci beaucoup pour l'invitation de parler aujourd'hui. Bienvenue à Washington, D.C. Et c'est difficile pour moi parce que chaque fois que j'ai écouté la même chose en anglais. Okay, I'll switch to English after trying to say thank you in French, but it's indeed a pleasure to be here and know that many wonderful colleagues are part of this and this very important effort set up by ENCA. So I'm going to talk about integrated approaches to studying the etiology of pediatric cancers. There are many new tools for important questions, but the real tough question that every parent asks every pediatric oncologist is, why did this happen? And we know a lot about how cancers developed, and you've heard very elegant discussions the last 2 days, and you'll hear more. But the question of why and what is the actual etiology is still a very difficult and challenging set of questions. And I think some of the new tools and big data are very helpful, but it really does take very deliberate investigation. So we know with pediatric cancer in this age, we have many different tools. I'll touch upon a number of these, from descriptive epidemiology to genetics and biomarkers, family studies, cancer genomics, environment, occupation, radiation. We know that pediatric cancers arise for complex reasons, and it's very rare that we can point to one particular thing. So we really need to look at the context and see that we know that there is germline genetic variation, which each and every one of us are born with. There are millions of variants in each person's genome. That's what gives diversity to the human population. And it also sets the stage, or the carburetor, so to speak, for how each individual responds to environmental exposures. And what those interactions are like have very profound implications for the somatically acquired mutations, whether they're in adults who develop lung cancer after smoking or melanoma after UV light. We now understand a fair amount about how those events take place. But when it comes to children, we understand less of that. And I think some of the key questions that have come out of the last 2 days are really, what are those dynamics of those changes in our genomes? How quickly does this happen? it does happen quickly, but to be able to measure that is really a very challenging question. And what are the trigger points for what clearly is a major theme in pediatric oncology now? And that is the disruption or really the disruption of development, whether it's plasticity or whether the developmental pathways, epigenetic pathways and the like that are changed. How and why those are changed, the question is, what are the environmental exposures? What are the elements due to bad luck? And what are those that are due to the somatic mutations that have already been acquired? And why does this happen faster in children? And why are there, in some ways, stronger drivers? Because so often in pediatric cancer, you may have just one or two events that are clonal. And I'm going to come back to this when I talk about thyroid cancer in children who were exposed to radiation following the Chernobyl accident, where we really do have an opportunity to look very specifically at very known, well-characterized environmental exposure, and then see how the cancers actually develop. Right. So established risk factors for pediatric cancer. We know that congenital anomalies and chromosomal abnormalities are clearly associated with risk for pediatric cancer. Family history, germline genetics, I'll touch on each of those. Ionizing radiation, I'll come back to that. Both sustained high environmental is established, not very low. And radiation therapy with respect to second cancers remains a continued problem in the aggressive treatment of children with the risk for second cancers. We know that paternal age is a risk factor for pediatric cancer, and this is a rather uncomfortable, difficult subject as we see the demographics in Western Europe and the United States move to older and older parenting. There is a small but real calculable understanding of that risk of an increase for pediatric cancer. Similarly for autism and other diseases, partially due to de novo mutations. And then, of course, select pharmaceuticals, such as the DES experience in the United States in the 50s and 60s. It's really, really the iconic, unfortunate situation. So if we look at birth defects, we can look at big data. And so Philip Lupo and Sharon Plon 2 years ago published this very impressive study where they looked at 10 million children born in the US. And we already knew that historically, things like trisomy 21 and acute leukemia were clearly implicated together. But what they could see is 1 in 33 children are born with birth defects in the United States, probably even more with new prenatal technologies, ultrasound, multiple high-definition ultrasounds being done during pregnancies. I know 2 of my 3 grandsons have all had incidental findings from ultrasounds that were identified. But when they looked at 10 million children, they could see that the presence of a chromosomal abnormality led to 11.5 times more likely the diagnosis of a pediatric cancer. For non-chromosomal birth defects, 2.5 times more likely. And if there were 4 or more defects, that was almost 6 times more likely. So this is telling us that there are these events that are probably occurring very early after conception. And what ones drive towards cancer and what ones don't is really a very interesting place to study. And we know that hepatoblastoma and neuroblastoma are the most commonly associated cancers, but we also know that there are many specific associations between specific defects and certain types of cancer, kidney defects, Wilms tumor, and the like. We also know, as I mentioned, in population-based analyses that parental age does confer a small but measurable risk of increased pediatric cancer. So the evidence suggests that the older the parents, when you look in 5-year blocks, so to speak, that it increases a small amount. And this is looking at hundreds of thousands of individuals in the population-based analyses in California. The Scandinavians have seen something somewhat similar, maybe a little bit lower. But the point is age, parental age, appears to be an important factor that we have to keep in mind, okay? This is, I realize, a very unpopular issue, but it is one that we at least have to recognize that this is the data that we have in front of us. What we know has not been established as risk factors are specific dietary patterns. There are many small, underpowered studies that suggest eating too much broccoli, not enough meat or fish. I think that the whole world of trying to map dietary intake in such a specific unifocal, singular way has been so far really unsuccessful. Specific foods, we know that cell phones and Wi-Fi, there is no evidence of increase in brain tumors, and we know that the large European study is about to come out and confirm that. We've seen that in the large cohort studies in the United States. Some of the case control studies, small ones early on, had suggested this, but the preponderance of evidence really is that cell phones and Wi-Fi are not associated with pediatric cancer risk. And then certainly ambient radiation and radon in the population is not associated with that with pediatric cancer. These are things that come up periodically. So when we think about the etiology of cancer, we do think about genetics. We know what we can measure what people are born with. Environmental lifestyle, we're much less effective at being able to measure those things, but we do have some sense of those things, whether it's pollution, whether it's microbes, whether it's exposure to alcohol secondhand, cigarette smoke, radiation, quality of water, weight, insulin, metabolic status during pregnancy. These are all important lifestyle questions that are still open, that are not answered at this time. And we know that it's really that interaction between the host genetics and these environmental lifestyle activities that are responsible for part of the etiology of cancer. And we also know that there are errors in the genetics in the background that occur with great regularity that are predictable. Granted that they're not common, but again, the idea of chance is really a very important question, really of probability and how we define that. And then we know that the outcomes are clearly related to those somatic mutations, and how much the genetics and lifestyle influence those are still very much open questions and survivorship questions. So I've been very interested for a long time in this question of studying germline susceptibility to pediatric cancer. We know that it gives us insights into cancer etiology, how the germline informs the somatic. It gives us insights into things that have been identified by large population studies, such as going to cancer registries here in the United States and Europe. And Joe Framini and others described some 40, 50 years ago a substantial difference between ancestral history for individuals with Ewing sarcoma. Those individuals of European ancestry, anywhere from 6 to 10 times more likely to develop the disease, as opposed to those individuals of African ancestry. And there's some very interesting genetics that we're very much involved in with Olivier in Paris and others to try and tease that out. But that's been very slow, simply because these are not very common diseases. We know that we can develop risk assessment tools, particularly in the adults. And in pediatrics, that's a very interesting question. But the precision prevention in oncology really comes with familial cancer. So we know that some 8 to 10% of children in large centers, when we sequence their genomes, we can identify a pathogenic or concerning mutation that may be important not only for that child, but for siblings, but also for the parents. And then we also know that germline susceptibility to pediatric cancer can be very important for the choice of drugs and particularly survivorship issues. And those are ongoing questions right now. Now, for genetics, we think of now genetic architectures, so to speak, of susceptibility. Cancer is complex. One gene alone does not cause cancer. There can be sets of genes, and there are different types of genes, whether in this figure showing that very rare alleles, very rare variants in the population can have very strong effects, like BRCA1 and BRCA2. And then we know that there are common variants that are implicated in diseases where we know that many together explain a good fraction of the disease. So for the rare alleles causing Mendelian diseases, we know about 125 different cancer syndromes. And many of these are described in pediatrics, very rare, unusual cancers, but mapping to particular genes that are very illustrative and give us new insights into particular pathways that are important for particular tissue-specific activities and pathways, so to speak. The genome-wide approach and now has actually many more, probably 1,400 loci and over 150 in pediatric cancer alone. And so diseases like Ewing sarcoma, where we don't see highly penetrant mutations, we see a number of GWAS loci that are a little bit stronger than the adult and can begin to explain a fraction of the risk for that disease. And then we know that when we look at the science behind these, that the cancer syndromes are usually due to very damaging drivers and oncogenes and tumor suppressor genes, whereas the genome-wide association studies are really perturbations of key pathways in ways in which the genome is responding to environmental stimuli. So when we look at the pediatric subset of these, we're still very early in trying to put these maps down. We do know that next generation sequencing has been very, very informative. As I mentioned, some 6, 8, 9% of children with cancer in large centers such as St. Jude's, who published that first very important paper in New England Journal of Medicine, and now a number of other studies have shown this, even in such as the Childhood Cancer Survivor Study here in the United States. We sequenced 5,500 survivors of cancer 5 years or more, and we could see about 8% had known pathogenic mutations that had been identified previously and registered in ClinVar and ClinGen. These are differences by cancer type, but this is really, again, important for the family history, because sometimes these pediatric cancers are sentinel events that the parents themselves are young and just entering the risk period for BRCA1 and 2 for breast cancer for mothers. This has been raised in diseases like neuroblastoma and in leukemia. Again, they're scattered among a number of different genes and a number of different diseases. So this still represents a small fraction of pediatric cancer overall. And so the landscape of these genomic alterations in childhood cancer is really important in how we map these back and forth. So we look at different diseases. So we've spent a lot of time looking at osteosarcoma, pediatric cancer of childhood, adolescents, and young adults. And we sequenced some 1,200 cases. We were quite surprised to find that literally 1/4 of the cases had a pathogenic variant in their germline. TP53 was the most common one, but there were a whole series of other ones. So raising the question of some pediatric diseases have probably a much higher fraction driven by rare pathogenic variants versus others. And the question is, when and where do we start consider screening or using these in ways in the clinic? And those are ongoing questions. So we know that we can get insights into how cancers form with small effects that are both common and rare strong effects, really following the Knudsen hypothesis of knocking out 2 copies of the gene, one in the germline and one in the somatic, cascade testing in families. And at this point, we're really inadequate in our data for polygenic risk scores and population prevalence of pathogenic mutations. I think some of the large sequencing efforts in biobanks are very informative in teaching us and really illustrating what are the distributions of these alleles. And then we can begin to really look at the cost-effective strategy based on the reduced cost of sequencing and how we would follow up these particular individuals. So again, coming back to the question of the etiology of cancer, now I want to turn to really where genetics and the environment are really colliding. So what we know about our exposures, there's a lot from the Dahl and Pito days and their assessment of causality of adult and pediatric cancers. We know a lot about somatic mutations. The Cancer Genome Atlas and the International Cancer Genome Consortium were terrific at laying out landscape analyses, but they paid virtually no attention to the exposures and environmental drivers, as well as ancestral backgrounds. So we're paying a price now, having to work backwards to try and understand what we can from those studies in terms of what's the relationship between the environmental exposures and the development of those cancers. So it's an incomplete and expensive endeavor. We can use things like mutational signatures and epigenetic profiles as forensic tools for epidemiology, but we're still rather naive in doing this because we need larger data sets to be able to ask the question, what are the endogenous processes that are going wrong? What's that within the cell? And what are the exogenous? What are the effects of particular environmental exposures that are driving particular patterns? So my key point is, going forward, we should never, ever think about doing cancer genome analyses unless we know something about the background, if we know the context of that cancer genome analysis. If we're going to understand etiology, we need to understand what the exposures are that were behind and leading up to the cancer genome analyses, if we're going to be able to approach the etiologic questions. I fully understand and support the importance of understanding what we can to develop new targets and ways to treat going forward, but there are related but not completely overlapping questions here, okay? So we have spent here in the National Cancer Institute over 30 years following individuals who were exposed after the Chernobyl accident, and particularly looking at pediatric cancers, and particularly thyroid cancer. So I'm going to talk about a paper that we recently published in Science in which we knew a lot about the environmental radiation exposure. We can look at the alterations in the cancer and for the first time really map environmental exposure with actual genetic events and trying to understand how and why and the timing of those particular cancers. So we all know that on April 26, 1986, there was an accident in one of the reactors in Chernobyl, spewing a radiation cloud that settled over southern Belarus and a good part of Ukraine, as well as in parts of Russia, with higher doses. And our work really is based here that I'm going to talk about in the Ukrainian cohorts, where the residential exposure led to the fallout of particularly I-131 in the community, in the grass that was then consumed by the cows. This was 1986, so there was not much in the way of nutritional options for families. And so the kids drank lots of milk that was contaminated for a period of time. So the model that we see is the weather patterns spread the radiation into the environment. The cows concentrated in the milk, and then young children drank that milk and had I-131 exposure. We've known for quite a while that I-131 can be associated with risk for particularly developing thyroid cancer. This is something that we've certainly seen in both the Chernobyl cohorts and also in other studies following Hiroshima and Nagasaki and other exposures to radiation. Certainly the second cancers literature has also shown this, that the pooled analysis, the younger the age of the child and the exposure to radiation, the greater the risk of developing thyroid cancer, particularly papillary thyroid cancer. So we went to the Chernobyl Tissue Bank, led by Jerry Thomas in the Imperial College in the UK, and were able to obtain 440 confirmed cases of papillary thyroid cancer, fresh frozen material. We had about half of them had non-tumor thyroid as well as blood. And then we did something like the ICGC and the TCGA, where we did very careful pathologic evaluation. These had already been seen by 6 international pathologists who were part of the Chernobyl Tissue Bank. We then did RNA, DNA sequencing, expression, epigenetics and the like, and for the first time really being able to do an integrated analysis where we know exquisitely the dosimetric exposure, how much radiation each and every one of these cases had. And so we were able to really look at the effect of radiation on the genomic features themselves. So this is one of the first snapshots of trying to understand how environmental activities and environmental exposure leads specifically to a particular type of pediatric cancer. So everybody who does these large studies does these landscape plots where they show the sex and age, but then the mutational counts, the different types of signatures, the different types of structural variants and driver mutations. But here we're able to do this and really segregate this on the basis of unexposed. We had 81 cases after the Chernobyl accident in regions nearby, but they were not exposed to radiation. And then 1 to 99 milligrays, all the way through greater than 500. And we could see that the characteristics of the driver mutations shifted from being predominantly point mutations, as you see in the red, to fusion proteins in the blue as you went to higher and higher doses of radiation. So this was a very important observation. We know that the mutational burden in these cancers is very similar to what we see In pediatric cancer, even the adult TCGA was on the low side, 2 to 3 orders of magnitude less than what we see in smoking or UV-driven cancers. And in fact, the Chernobyl was even lower. So in terms of single point events, radiation did not appear to be inducing many single base events. And that's important. When we started looking at the different signatures for single base, we saw nothing. But when we started to look at small insertions and deletions, we could see that those ratios started to look very interesting. So if we did the ratio of insertions and deletions to the SNVs in the background, we could see that the SNVs developed steadily in the background with age and with radiation. So the effect of radiation on small deletions substantially increased with the dose in radiation. Increasing radiation leads to more small deletions, and we'll talk about how we can see that when we then normalize that with background SNV ratio, because almost all of the single base changes we saw were due to the clock 1 and 5, the steady progressive errors that take place with genome doublings. We could see that that effect was even stronger. This is a finding that was interestingly in parallel, published shortly after our paper came out, in looking at individuals who had had radiation and looking at primary and recurrent gliomas. Their radiation exposure, and particularly small deletions, were enriched in the same cosmic indel signature 8, was exactly what they saw, similar to what we had seen in our cases. Now, we had protracted longer exposure. Unlike the radiation exposure of a therapeutic radiation, ours was over weeks to months. So it's a very interesting thing. When we looked at the driver mutations, in which we were able to identify 98% overall, 93.5% of them were in just a handful of genes in the mitogen-activated protein kinase pathway. We knew that this was important from TCGA in thyroid cancer in 60–70%, but here in the pediatrics, we saw it exclusively there. And so when we looked at the radiation exposure, the increase in fusion drivers, increases in indels, the structural variants had an increase also in relationship to radiation, really suggesting that what we knew from animal studies, that double-strand breaks are very important. But it's that non-homologous end joining mechanism that seems to be very important. So when we look at DNA double-strand break repair mechanism, there are many different ways that this is done. And so we could take the RNA data from the fusion proteins, take the DNA data as well from the same case, and look at the specific joining of 2 different regions and ask the question, are there characteristics that would suggest a particular mechanism that had been disrupted by radiation? And the answer is yes. We could see that when we looked at things that were more than 5 base pair deletions, and we could look at the microhomology really strongly suggesting that non-homologous end joining was the primary way in which the fusions and the SVs and probably the smaller deletions were being generated. So it really does fit with what the animal data had showed us before. So we looked at this, and then we said, all right, can we see the detritus of radiation in any way? But methylation patterns in these cases were driven exclusively by the drivers and not by the radiation. We could not see an epigenetic pattern that suggested radiation versus non-radiation in tumors. Similarly, same thing for RNA expression. It was all driven by a BRAF or a RAS. And so the driver gene takes over in these cancers. We could not identify a specific biomarker. So in summary for this study, we went from the event to the radiation exposure to DNA double-strand breaks that we could see in the thyroid cancer, to a landscape analysis that really has shown us some very important subtle issues related to radiation dose and DNA double-strand breaks. And we're now deep into studying the normal tissue that was irradiated as well in these individuals but didn't develop cancer. And we see very low background events, but they, again, show this double-strand break. We don't see the single-strand events that you see in many other environmentally driven cancers. So our key findings are that a high fractioner in the MAP kinase. So there's a parsimony of this particular pathway, similar to what we've heard about in some of the other cancers, that they're developmentally very important places that get disturbed. And we know the thyroid is really a sanctuary that has a very interesting development with respect to stem cells. So it's exquisitely sensitive to the radiation, and particularly I-131, those increased double strands that we think are through non-homologous end joining. And the epigenetic and gene expression profiles are defined by really the driver mutation. And we don't see strong field effects. So we can't identify whether somebody was exposed or not exposed to the moderate levels of radiation. We're going much deeper with the FFPE tissues now, because we have double that number to be able to look at younger individuals with higher doses. We can see the multifocal diseases and asking the question, are they synchronous or not? In metastases, we've just about finished that analysis, and we see there are a few new metastatic features, but we don't see them as different diseases overall from the primary. And then expanding to other radiation-exposed tumor types is really important. There have been many wonderful people, Lindsay Morton and Danielle, and our colleagues, academician Tronco and Gadi Getz, Jerry Thomas, have been marvelous. So I'm going to spend the last 3 or 4 minutes talking about how important, as we think about these pediatric cancer questions, the FAIR principles are, and data stewardship, that things have to be findable, accessible, interoperable, and reusable. So here at the NCI, in the last 2 years, there's been a presidential initiation of a $500 million effort called the Childhood Cancer Data Initiative that will look at different data types and has as the goal to set a community standard to be able to bring together many different people, from advocates, families, hospitals, networks, to sharing data to improve treatment, quality of life, and survivorship, and trying to understand the etiology of cancer. So this is building a federated infrastructure here in the United States that we fully expect to share and work with our international colleagues, opportunities to make data more available, maximizing every opportunity to improve what we can in treatments and outcomes for children with cancer, and enhance and develop new tools and methods to extract data and knowledge from that data. So this is a major effort that's here in the United States, taking things such as the Children's Oncology Group Project Every Child, the St. Jude's Cloud, the PEDS CBI Bioportal, Treehouse, the Target, and now bringing new activities such as National Childhood Cancer Cohort, which I'm one of the coordinators of, and the Childhood Molecular Characterization Protocol, a way in which we really have every expectation of more integrated, larger analyses to be able to get more refined. This is going to be particularly important for some of the rarer types of pediatric cancer. We know pediatric cancer is already relatively rare compared to other adult cancers, but this is an effort that really is trying to unite and bring together here in the United States a central childhood cancer data platform that will link out. So here's our structure, with cross-cutting issues, but particularly looking at the childhood cancer data platform and the molecular characterization protocol. And that is a program in which we know that of the 16,000 children diagnosed every year in the United States, we have as an initial goal to reach 4,000 to 5,000 who would not normally have access to molecular diagnostics, particularly in underserved areas, with a particular focus on the pilots on sarcomas and central nervous tissues, tumors, and rare cancers here in the United States, to try and set standards so that every child diagnosed would have appropriate clinical and molecular characterization from the start. So again, the tools are here. It's time to move to the next gear and be able to get at pediatric cancer. So my final important points are les points importants, because the causalité est très difficile à déterminer maintenant. Environnement et exposition très difficile à distinguer cause et effet. Et des grandes études sont nécessaires. Et les cancers pédiatriques sont des malades rares. L'ensemble partage des données étendues. C'est très, très important. Nouvelles technologies nécessaires pour identifier l'exposition nombreuse. Et la coopération est fondamentale, nationale et internationale. Et merci beaucoup et bonne journée.
Thank you, Stephen. We really appreciate your last slide in French, actually. So we have many questions from the chat, but I will start with one question. I would say it's a naive question. We know that germline genetics will define around 10–15%. It basically will define, yes, 10–15% of cancer. But from the study, for example, from St. Jude and etc. But every year, basically, I think we can detect, like the study from DKFZ with the ELP1 mutation in the sonic edge group of medulloblastoma. So it's been that, do we think that looking deeper in the genome, we will be able to basically increase the percentage of potential germline mutation in pediatric cancer?
Absolutely. I'm going to say something a bit controversial that Joe Farmani once said, that cancer is 100% genetics and 100% environmental. So I think that it's easy for us to understand the 10 or 12% of those highly penetrant mutations. I think that we will find more in those rare circumstances that will be more of the tail. But I think common variants are very important, such as we see in ALL, in Ewing sarcoma, in neuroblastoma, where John Maris and others have shown that in combination can explain a part of it as well. And again, the genetics set up the opportunity for the cancer to develop. But what triggers it? And is it due just to that genetics or the interaction of those genes with the environment? That's, to me, the question of the next decade, to understand how and why all the very elegant things that were described in the last 2 days in this meeting, the changes in plasticity and development, and epigenetic profiles, is that just by chance? I think that we can continue to decrease the number that we would explain by chance. How much germline and how much environment and that interaction really remains in front of us. And I think large integrated studies are going to slowly but surely explain more and more of how we understand that. Okay.
Thank you. So I will link to this to this question. I will take a question from the chat. It is about 10% of pediatric cancer presenting a germline mutation. How much do we know, or do we think genetic susceptibility may cooperate or contribute to the tumor formation?
That's an excellent question. How tumors form, you know, again, we go back to the Knudsen hypothesis, which showed in retinoblastoma that a germline mutation was one of the first event, and then a somatic, a change in the actual cells in the retina that had that knocked out the second copy of that gene, and then retinoblastoma would develop. I think tumor formation is a really important question. Does that occur suddenly or over a period of time? So our experience in Chernobyl is we know that those children were exposed for weeks to a couple months. So that's when the radiation that they saw that cut and generated the fusion proteins and the SVs and the insertions and deletions. So we could map that directly to that environmental exposure, and particularly the higher exposed children. The question is the lower exposed, you know, there are some background sporadic cases that had nothing to do with radiation, and how we sort that out, again, is a more challenging situation that I think numbers, larger studies will help us to explain more and more of the fraction of what is radiation driven and what is in the background, which is occurring for other reasons, smoking, environmental, you know, genetics, you name it.
Yeah. A question also I think is quite important looking at your slide and your data is, the question is, it is known or suggested that increased risk associated with parental age is due to an increased number of mutations in germ cells. So do we know that?
Well, that's a great question. So we published the second paper about Chernobyl that I didn't talk about. It's looking at this question of de novo mutations. So we know every time that a conception takes place, there are anywhere from 40 to 70 errors that take place in the ova and testes, the sperm, and so that you can see these things that were Mendelian inconsistencies that are not seen in the germline of the mother or the father, but they arise in that context. And so the de novo mutations account for a fraction of autism in individuals, particularly older parents who have autistic children. And one of the really interesting questions is we are yet to really map that in pediatric cancer. Are some of the, for instance, the osteosarcoma, we talked about those up to perhaps a quarter of osteosarcomas particularly under age 30 may have a pathogenic mutation. What fraction of those may be de novo mutations? And since we don't have, you have to have both the mother's and father's genomes to be able to make that determination. That's why I think family studies are going to be crucial to be able to start to understand the part of the mechanism there. So I think that, again, those germline errors that are taking place over time are very important and potentially critical factors in explaining part of that interesting phenomenon.
Yeah. So probably a last question from the chat as well. How do we overcome the GDPR issue? So basically sharing data and personal information. I think this is also an important question in this field.
I thank you for raising that. It is something that is utterly frustrating to us. We have 30, 35 studies that have been ground to a halt. I think it needs compromise on both sides. I personally think I can't blame it all on how the EU wrote its charters for that, but I think that that needs to be addressed at the EU level in rethinking the restrictions on academic sharing of information, in the same way that some of the United States tort laws that keep us from being able to sign those documents need to be reevaluated. I think there is a middle ground, and I know we here at the National Cancer Institute and at NIH are working very hard to try and overcome that, but much of that is the language in the EU, and that's something that I hope it would come out of a meeting like this, that advocates and politicians would say, we need to change that so that we can better share the information, because to solve some of the riddles of pediatric cancer are going to take internationalism. It is just too rare and too uncommon to accumulate enough cases to be able to make some of the insights and things that we need to sort out on just national or regional basis. So the GDPR does need to be taken on and does need to be changed, and we will do whatever we can to affect that. Thank you for that question.
So thank you, Stefan. I think we will move on, but we really thank you because I think your presentation and question and discussion were really important for not only scientists but also for parents and patients. So thank you again. So now we will move to the second speaker. So the second speaker is Peter Karchenko. So Peter leads a computational biology group at the Department of Biomedical Informatics at the Harvard Medical School. So his team focused on the analysis of intratumoral heterogeneity in different cancer types, as well as the interaction between tumor cells and the macroenvironment. And today, Peter will focus specifically on neuroblastoma and the use of single-cell technology. Peter, are you with us?
Can you hear me?
Yes, I cannot see you, but I can hear you. So welcome,
can you see the slide?
I can see the slide. So welcome,
Excellent.
Peter.
Thank you very much. So I'll focus on the single-cell applications, basically single-cell transcriptomics, and I'll try to cover generally how it's a research tool, generally how it's being used in analysis of cancer, and then I'll focus on a particular study of neuroblastoma that we've been carrying out with our colleagues. Okay, so what single-cell RNA-seq analysis allows us to do is to take a cell, a little dot, right, and get a sparse sampling of the transcriptome present in that cell, so the genes that are being actively expressed in that cell. Altogether, we're going after protein coding genes, so it's about 10,000 genes potentially that you could get from, let's say, a human cell. But in reality, we only get a small fraction of the molecules that are actually present in the cell, and so the information about the cell is a little bit uncertain. But ideally, it can tell us about what type of cell it is, what is it doing, how it's being influenced by, let's say, microenvironment, and so on. So while we can't really do much on the result of a single cell, if we sequence more of these cells, we can start to see some sort of patterns. In particular, we can recognize groups of cells that look similar to each other. And here, basically every point is a cell, and they're being positioned next to each other if they're similar in terms of their expression state. These analyses can lead to sort of clustering results where you could look at the expression patterns and actually identify what types of cells these are. And I put some labels here. So that tells us about the composition of the tissue, of the biological sample that we're looking at, whether it's a healthy tissue or diseased tissue and so forth. Now, if it is a dynamic process, let's say development, differentiation, or response to some kind of perturbation, you could sequence more cells, essentially analyze more cells, and perhaps catch some of these transitory states that connect different populations. I've shown them in gray here, and that kind of tells you, like an ant path, basically gives you a little bit of a trajectory within the space, transcriptional space in this case, and tells you about how the cells may be shifting from one state or from one cell type to another. So that gives us an ability to analyze and characterize transitions to some extent. You could approximate this, and here we basically will be looking at developmental related differentiation. So you can approximate these types of transitions as trees or paths in this expression space. And if you imagine a cell, in this case, this will be actually a neurocrest cell that's delaminating and going through differentiation process. You can imagine it following a path and then essentially choosing or somehow being pushed into one fate or another fate. And what we can monitor through these types of measurements are the genes, key factors that are involved in these types of transitions. To some extent, if you can guess the direction in which things are happening by looking at more detailed molecular patterns. So on its surface, so this is a very powerful tool for basically looking at cell composition, cell activity, and the transcriptional state is kind of a nice molecular modality to look at because it integrates obviously the state of the genome, both in terms of any aberrant mutations that are there that are significant, as well as epigenetic action, and it also integrates faster modalities related to protein signaling and so on. So it's a nice intermediate state to focus on. Now, since we're in the data management section, I'll mention some of these data management issues. The remarkable thing about these types of techniques that over the past 5-7 years have grown very significantly. So as any kind of growth trend, you have to compare it to Moore's law. So here on the x-axis you have years, on the y-axis this is log count on log 10 scale of transistors and different processors that we had. And here more interesting, so in red, in bold red points, I'm showing landmark studies, and what I'm showing now on the y-axis is the number of cells measured in these landmark data sets or publications. And basically over the years, from about 2010, when the best data sets had only a handful of cells, 5-7 cells, we're now gotten to a point where we can realistically measure millions of cells from a given sample and get a very comprehensive kind of statistical view of the transcriptional composition and the states present in that tissue. Small dots are all the other studies that are being published. So they're a little bit below the curve, but still going up. For reference, this is the number of cells in the human body approximately. Now, what this means is, on one hand, it's very gratifying. We're getting more information, but also we're catching up with a processor growth. That means every year is basically getting harder and harder to process and analyze this data on the existing computers. Right now, a typical study, for multiple data sets can gather anywhere between 100,000 and maybe 1 million cells. Even if you treat this carefully in some compressed or sparse manner, it's still a significant amount of data to analyze, especially given a variety of analysis that we'd like to do. More importantly, because these techniques are accessible and they're very nice commercial implementations of these protocols, they're being rapidly adopted by multiple labs, many research labs actually, and so they're also being put to work in realistic study designs. So instead of having sort of one-shot experiments where you simply take a first look at a tissue to see its composition, for instance, now you would have a cohort of individuals. You would have, let's say, group of controls being compared to a treatment and so forth. Furthermore, there are these consortium scale efforts to generate very large atlases, and I'll mention it in a minute. And there we have thousands of experiments and likely billions of cells, the culmination of these efforts. And so if we were to take advantage of this, this type of data, we basically need two things. We need effective computational tools that can essentially crunch some, the data sets of such volume, as well as we need additional statistical considerations, for instance, to make sure that the results are not being thrown off by inter-individual variability or certain biases within the cohorts being studied. So just to give you a glimpse at one of the computational problems here related to the example that I'll be showing. So this is an example of a study of a metastasis to the bone marrow, and basically what each square represents. Each square has little cells, again, little dots representing cells clustered, by subpopulations. So it's a separate measurement, right? And so in these tumors, in this study, we can analyze solid tumors from different individuals that have been surgically extracted. Where we can get, for instance, liquid aspirate of bone marrow and right next to the metastasis, so we get another set of samples. Distant vertebrate, for instance, as a control. We can have healthy controls again. So you end up having a number of samples, right? Quite a bit of data. And so far, every sample has basically been analyzed individually. And it turns out that, for technical reasons as well as genetic and biological reasons, whenever you look at the state of the same cell type in different individuals, it will be a little bit different. So first thing, what we want to know from these types of panels is for instance, what is the compositional impact of the disease, in this case, metastasis, right? Are there certain subpopulations of cells, states, or cell types that, let's say, increase in abundance, decrease in abundance? So that's kind of a compositional shift. And the other thing we want to know is sort of the flip side of the same coin, which cell types have changed significantly in terms of their state. For instance, there's immune activation or let's say dysfunction of certain immune subtypes. So we want to monitor these types of shifts, expression shifts within a particular cell type or subpopulation. And the first thing you need is basically a way of tracking the same subpopulations throughout this entire collection. And as I mentioned, if you simply put them together with no corrections, basically what happens is that you see these subpopulations that are defined by individuals, right? Because the cells taken from the same sample, the same individual, will be more similar to each other. So there are computational corrections that are needed to overcome this, and that's an example, let's say, of a computational procedure that's pretty difficult to do at scale still. This is an approach that we've designed in our group to do this type of analysis. Basically, the idea is pretty straightforward. We'll be looking at pairwise comparisons, so we'll take a sample from one individual and another and do sort of preliminary kind of mapping between them to establish putative relationship between different cell types and subpopulations within these two samples. And then we'll do this for all the pairs. So as you might imagine, if you have a lot of samples, that will be a lot of pairs. But if, let's say, this red subpopulation actually represents a recurrent subtype of cells across the entire collection, it will tend to map to itself across these multiple comparisons. And that type of pattern is very easy to recognize statistically in this type of a joint graph. So we can use this approach to basically annotate the subpopulations within the entire collection, as well as propagate information between them. So just at the risk of generalizing to kind of categorize, where I think how these technologies are being applied. So a key effort is still on basically mapping tissue composition, whether it's normal tissue or diseased tissue. And there are a lot of consortia involved in doing this. So probably most known one is Human Cell Atlas. Here, this is kind of a large collaboration, let's say, of different labs and different initiatives, even that aim to put together a nice reference data set of human tissues. And there are related projects such as HubMap and Brain initiatives that are funded, at least within the US. And the other aspect, let's say, would be comparative analysis. So in this case, really the aim is a little bit different. This is where, for instance, you're looking at healthy controls versus cancer tissue or treatment, versus control. And what you're looking for is potential targets. Realistically, you're looking for cell types that are most affected, as we discussed, either by their abundance or by their state. And then because you have such detailed molecular profiles of these cells, you can actually generate hypotheses as to what particular pathways are responsible for these phenotypical changes or cellular changes, and try again to generate follow-up experiments that would try to test some of these targets. And here, we have in US Human Tumor Atlas network that is aiming to do this, and there are quite a few disease-specific consortia that have spun up. Just to expand a little bit further, just in terms of these different types of applications. So we talked about mapping, talked about assessment of the disease impact. In relationship to cancer, also particularly treatment, the obvious design is to look at, let's say, minimal residual disease or cells that are resistant to a particular treatment, and again, trying to understand the molecular signatures that are associated with that. To some extent, there have been some work suggesting that eventually these types of single-cell approaches can be used for more diagnostic utility, such as detecting rare cell types. I think it remains to be seen. So the study that I'll talk about today from our group and our collaborators is really of the first type. So we're really looking at kind of initial view of neuroblastoma with some of these very high resolution techniques. So this example that I was showing you is actually mouse neurocrest development, and that's what we've been studying for other reasons, just for developmental reasons. And what you have here are neurocrest cells maturing, and then going through a series of cell fate decisions, including this last one that essentially goes from this kind of a bipotent progenitor that apparently exists. It was a bit of a surprise into autonomic phase or mesenchymal phase. And this was immediately kind of reminiscent, although it's a distant analogy, to the heterogeneity that was characterized in neuroblastoma the year before by a couple of groups, including Olivier and University. So we were interested in whether there are actually more closely related parallels to the heterogeneity and potential transitions between these types of mesenchymal and adrenergic populations that we could actually see in the primary samples. So we've teamed up with a group of Nina Varjano and Karolinska Institute, a young PI, to look at primary neuroblastoma tumors. These are from surgical samples and biopsies. Neuroblastoma is a pediatric cancer of neurocrest lineage. The exact sort of cell of origin and the path of tumorigenesis is still, I think, a subject of debate, but they are highly heterogeneous tumors. And in this regard, being able to view them at this kind of single cell resolution seems to be a benefit. The design is pretty straightforward. As I mentioned, for collecting these surgical samples, the big advantage is that we have an opportunity to actually get them fresh. So all these single RNA-seq techniques at this point are rather sensitive to the state of the tissue. There's no easy way of kind of fixing sample and just keeping it around and then measuring it later. The time between the acquisition of the tissue and processing is really critical and should be down to like an hour or that order. So we're able to do these kind of dissociations and then measuring individual states of the cells. In parallel, we're also measuring copy number alterations within these tumors. So altogether, we have about 20 patients, a bit more at this point. We're doing these assays on them. So here, every square is, again, a patient, and this is the integrated view. So using these integration tools, we basically put together data from all the patients, and then we try to classify the subpopulations of cells that we see there. This is how different samples map out to this general space. So using, in this case, computational techniques that I'll detail in a bit later, we try to predict where are the malignant cells within this picture. And what these analyses suggest is actually a fairly broad presence of malignant cells in the adrenergic compartment, which was expected, a little bit in the mesenchymal compartment, and also this kind of a connection in between that I'll really focus on. So I should say that the exact topology of these types of clusters is well, that's something that needs to be tested, right? So as we take this high-dimensional genomic data, reduce dimensions, and project it, onto 2 dimensions so we can see it. Obviously, a lot of distortion takes place, and when the question comes up, like whether, for instance, this adrenergic population is actually continuously connected by a bridge to a mesenchymal population, so we have to do quite a few additional tests to ensure that it is actually something that we're confident in. In this case, we've gone through these types of tests to make sure they're not doublets and so on, and actually it seems quite robust. Okay, so let's focus on this internal population that is suggested to be malignant. In this case, so we see, let's say in the mesenchymal subtype, very easily recognizable markers. The adrenergic population again has, kind of classic markers that we expected. And this population in between, so when we started looking at just the genes that actually mark it, it was very clear to us that it actually looked more like Schwann cell precursor cells or something related to sort of early neurocrest state. This was a bit of a surprise, so we actually started digging into this and really spent a lot of time trying to characterize this SCP-like population that appears to be connecting the mesenchymal and adrenergic compartments. Okay, so the first thing we can do is actually take some reference data from healthy tissues, from developmental reference data, in this case from adrenal gland as well as kidney, and try to establish more precise correspondence of different cell types seen in these neuroblastoma tumors and normal tissues. So what I'm showing here is the same embedding. So it's the same kind of expression space into which the cells are projected and here you have cells from neuroblastoma samples. This is the large adrenergic population, for instance. And here we have cells from the adult adrenal sample. So from the adult adrenal, we do have some mesenchymal populations obviously as well. So they map, at the same position with the mesenchymal populations in neuroblastoma. Mesenchymal cells can be, subtypes can be difficult to distinguish, so that's expected. Here, some chromaffin and sympatoblast cells. Sympatoblasts break up into a couple of subpopulations, but all of them are placed within the same general area as the adrenergic cells in the neuroblastoma. So what that tells us is that these are the closest matches, let's say, most closely closest analogs of the adrenergic cells. The SCP-like population, this bridge population we were surprised by, is actually co-aligning with the SCPs from the adult adrenal cells. And the same thing, if you look just at expression correspondence, there seems to be a nice match. Now, they're not exactly the same. So they share a large fraction of the expression state. So here in this column, we show adrenal SCPs, and here we show these neuroblastoma SCP-like cells. And these are other cells in these samples. And so you see that, they share, for instance, prominent expression of S100B, PLP1, SOX10, and so on. All the markers we would expect from SCPs, but there are genes that are expressed in the normal adrenal SCPs that we don't see, including FOXP2, which is an adrenergic transcription factor in this case. Also, there are genes that are expressed in these SCP-like cells that we don't see in normal tissue. So they're a little bit different, but they seem to be the closest analog of SCPs anyway. One thing I should point out here is that they all, among these shared genes is ERBB3, which is a receptor for neuroglin-1 and RG1 gene, and that normally confers a dependency on the Schwann cell precursor cells. So the Schwann cell precursor cells migrate along the nerves during development, and that is the signal that essentially ERBB3 and RG1 that maintains them on the nerve, ensuring their survival. So the surprising thing was, again, looking at these neuroblastoma tumors, in this case staining for NF200 neurofibers and SOX10 for these CP-like populations, that in most samples, these are different patients, in most samples, the vast majority of these CP-like cells are actually localized close to neurofibers. They're not fully fledged nerves. You could see them elsewhere in the tumors, but they still express in RG1, and so apparently sustaining this type of interaction. Now, a key question, of course, is whether they're malignant, and there's early literature, obviously, an observation looking at sort of non-malignant Schwann cells that can infiltrate the tumors. So this was still is a big point to analyze. And so there are several ways in which we could do this. So we could look at the expression patterns using tools such as inverse CNV or similar statistics to basically monitor for increased blocks of expression or decreased blocks of expression along the chromosomes indicative of the CNVs. And so we can score different populations using these types of techniques. And again, what we're looking for is this adrenergic population that is sort of classical neuroblastoma tumor cells having high scores, indicating a large number of these CNVs there, as well as in some of the tumors, at least prominently, these SCP-like cells showing this inverse CNV signal as well. Now we have the CNV data, actually, genetic CNV data on these tumors as well, so we could look at these specific regions and look for allelic disbalance, which is a slightly more robust signal, although more sparse, indicative of these amplifications or deletions. And again, here, just statistically testing these different populations for the same pattern of allelic change that we see in the adrenergic cells. We see that in this SCP-like and in some cases in mesenchymal populations, there's a notable signal suggesting presence of these aberrations. Of course, these are RNA-seq data. This is only certain parts of the genome are seen in terms of mutations. So very low coverage and rather noisy. So additional confirmation, of course, would be needed. The most direct way we could do it is using DNA and RNA FISH. Here using ppm-M1D, which is on 17q amplification to look for the number of copies of that gene, basically, and as well as SOX10 markers for the SCP-like cells. And what we see is basically that there's a fraction of these SCP-like cells that appear to have aneuploidies, in this case, amplification of 17q, again suggesting that there are, or indicating that there are malignant. There are some concerns. One thing we were surprised is that we didn't clearly see a subpopulation of SCP Schwann cell precursor cells or Schwann cells, or mature Schwann cells, that would be non-malignant, right? So in terms of transcriptional state, we would expect to see sufficient difference, so that cluster would essentially be split into two. We don't really see that, although our data indicates that it's only a fraction of these SCP-like cells that is actually malignant. So what we've been working on lately is actually, in collaboration with Martin Enge at Karolinska Institute, is combined DNA and RNA-seq analysis at a single cell level, and in fact have some initial data confirming aberrations in these SCP-like states. So going back to this kind of crude analogy with a mouse, so here we have this neurocrest sulfate decision going from migrating neurocrest cells into either adrenergic or mesenchymal parts. And so the analogy is that this transition bridge in neuroblastoma might be a kind of a similar structure. Of course, that brings up a question of directionality. If these cells are malignant, are they actually serving as a source of, let's say, mesenchymal and adrenergic malignant populations, or are they serving as an intermediate point, perhaps in this type of plasticity transition that can take place? So we could try to answer this with some of these, again, computational techniques that try to predict directionality of the expression change in this case by looking at intronic exonic splice patterns. It's a very weak signal. It does suggest that it goes into both directions, but of course that would need to be validated. Ultimately, we would to actually confirm this, right, we would need to screen a sufficient number of patients to see how the repertoire of genomic aberration changes between these three populations. In this one sample that so far we've been able to get detailed single-cell DNA data on, it does seem like the STP cells appear to be an early precursor of these malignant populations. One thing I should mention is that the whole picture is very much dependent on mechanism-amplification in a variety of ways. So, for instance, this NRG1 ERBB3 signal that's characteristic of SCPs and appears to be active here. So you could see, very, very notable difference in survival, in tumors that do not express either NRG1 or ERBB3. There's a significantly worse survival. But perhaps more importantly here, we separate the samples into MECAN amplified and non-amplified. And we look at the expression relationship between this NRG1, the ligand expression, and the receptor expression. And what you see is that for MECAN amplified tumors, essentially, they don't seem to have dependency on that. They can have different range of NRG1, generally not much ERBB3, and the survival is obviously poor. This is, again, going back to the survival question. So in general, we see the SOX10 high, this is going back to bulk data, SOX10 high tumors that would presumably have more of these SCP-like populations having better survival. But the key confounder here is that actually SOX10 expression in the MECAN amplified tumors is actually quite low. So altogether, it does appear that the MECAN amplified tumors play by different rules in this case, and so we would, going forward, would probably want to analyze them separately. There are other observations that we can gather from this type of data. For instance, the heterogeneity within the main adrenergic compartment. And again, here, we're aggregating data from about 20 patients, right? So the only types of patterns that we see here separating between different populations are the ones that are recurrent between multiple individuals, right? So we're looking for kind of common trends of intratumoral variation within these patients. The main findings, observations, at least, are rather straightforward. We see this kind of more mature adrenergic subpopulations next to less mature one that's notably marked by SOX11, as well as actively proliferating population. Now, the interesting part is overall across patients, what we see is that the abundance of that SOX11 in mature population is well correlated with the abundance of that proliferating population, suggesting that it contributes directly to the mature subtype. There are, again, we can go back to bulk data to try to assess association of clinical variables. In this case, simply looking, let's say, at the SOX11 expression, and its highest expression is in stage 4 neuroblastoma, but not in 4S, for instance, which regresses by itself. So most severe stage that does seem to be correlated with this presence of these, increased presence of the SOX11 population. Again, in terms of survival, this SOX11 high population on average has notably significantly worse survival. But it should be noted that if we go back again to the MECAN amplified patients only, we don't really see a difference. Again, suggesting that in this mechanism as well, the MECAN tumors are able to sort of play by their own rules. Okay, and I'll move on to summary. So this is an example of essentially a mapping exercise, right, for neuroblastoma. In this case, collection over 20 patients using the single-cell RNA-seq techniques. We're trying to see the composition and the detailed transcriptional states involved in these tumors. There's a high degree of inter-individual variation, right? So you could see this in some of the slides that I've shown, where we see individual patient data. And that really kind of goes back to this whole big data question, because on one hand, we are generating very large data by our sort of world amounts, right, references. But realistically, when it comes to being able to integrate across variation, or different subtypes of tumors, that's really not enough data. There's only 20 individuals. Obviously, look at genomic studies where you have thousands of individuals. This is probably the types of numbers that we need to be seeing in order to start generalized conclusions more broadly. We see multiple subpopulations of malignant cells. That was perhaps the most interesting part, including this surprising SCP-like population that does look like Schwann cell precursor population with different signatures, including the CRBB3 dependence, NRG1 dependency. Depending on how lineage tracing and attempts or analysis will work out, this potentially could be an early precursor population. It could also be a transitory intermediate, although we, at this point, favoring this first hypothesis, at least based on the data from a handful of individuals we've analyzed genetically. Within the adrenergic compartment, we see this prominent SOX11 immature subset that correlates with poor prognosis. And again, what I wanted to highlight, that it's very important to stratify populations in these studies. Of course, that's been well known. But even if we have 20 patients, separating subpopulations related to, let's say, MECAN or other driver mutations is very important. And overall, the scheme that we sort of have in mind is that this is under normal development, where we have these neurocrest cells migrating through the body early on, and then later on migrating along the nerves as Schwann cells. The hypothesis is that some of these Schwann cells early on accumulate mutations that predispose, at the very least, or serve as early clones or progenitors of these malignant populations, both adrenergic and mesenchymal, that they're able to seed. We cannot really rule out that there's plasticity going directly between these adrenergic mesenchymal populations, although we don't really see a separate route other than through this Schwann cell precursor population. And with that, I importantly wanted to thank my collaborators. So, of course, Nina Mariella, who has been leading this effort on the neuroblastoma measurement, Igor Demeka from Karolinska Institute and Vienna as well as a group of collaborators who are helping us to scale this up, including Olivier Delattre, Shazam Shlesia, Martin Enge, as well as a number of postdocs in the institute who have been working on this, Tale Olsen in particular, and Shannon May. Thank you very much. I'm happy to answer questions.
Thank you, Peter. Really fantastic presentation. So we have many questions from the chat, so I will try to classify them. So first, some question about, I would say, the technique. And so the first is, in your neuroblastoma studies, the identification of the tumor cell is done through some kind of score, if I understood correctly. Is this score exactly the same independently of the genetic alterations, or it needs to be adjusted based on the genetics?
Yeah, so that score, essentially, it tries to detect the presence of genomic aberrations. We can only detect very large ones. So let's say large amplifications, duplications, or deletions of parts of a chromosome. And there are two sources of evidence for this. One is that, on average, if you amplify a region, it tends to show higher expression levels, right? Higher activity. That tends to be a rather noisy signal. And the other evidence is that, in terms of the actual code, the sequence that's expressed, right? If you have heterozygous alleles within that region, you will see a disbalance in terms of allelic ratios. That is a much more robust signal, but is sparse. And so those two types of signals are primary ways that we're currently using to infer these aberrations or recognize these malignant aberrations in the tumors, in these RNA-seq data. It can work in principle for any combination of aberrations. But if there are very few aberrations, for instance, very few amplifications, or they're very small, the sensitivity of this approach is poor. That's why we're trying to complement it with additional validations using orthogonal techniques and eventually DNA sequencing.
Okay, thank you. So another question that is probably linked to the first one is, do you have some explanation for the presence of this SCP-like population in your data as compared to some other recently published data that failed to identify those cells?
Yeah, so that's a great question, and obviously, we've been talking to these groups. I'm actually, I must compliment neuroblastoma community on generally being very open and willing to discuss data that hasn't even been published yet. We think that a key difference, well, there are two sources of variation, let's put it that way. One is technical, right? So, we were fortunate enough to have this kind of workflow to measure fresh tissue, and the dissociation of the tissue and the treatment of the sample is extremely important for these methods. If the protocol is a little bit different, the cells, some of the subpopulations may be essentially not visible because they were not extracted, or that they don't survive this dissociation protocol. So that's really one key kind of characteristic that we're looking and discussing with other groups. That makes our data different, we think. The other one, to be honest, is inter-individual variation. So again, even we have a fairly extensive collection, 20 individuals. It's quite likely that if we measured 15 or measured a slightly different group, we would not be able to catch this because they're actually fairly rare, and then in some individuals they're present in very small abundance, right? And in a handful of individuals we see them being quite prolific.
Okay, and probably a last question or two last questions, and that's great because I wanted to ask this question actually. So, do you have tested for SCP plasticity and possibly a subtype shift, and also have you tested response to chemotherapy of these SCP cells?
Yeah, these are excellent questions, and this is obviously where we want to go, and with Ninib's group and with help of other groups have been working on this for over a year now. It is really challenging. So the main challenge is that these SCP populations, they're very difficult to culture. So as I described, they do have dependency on, for instance, neurofibers, and we don't have kind of a proper mouse model. If we're trying to create, let's say, a culture, cell culture, they don't survive in the culture. They basically don't propagate. Same thing goes for PDX. These types of transformations that are required for these models essentially don't carry them over at this point. There are much more complicated things that we're hoping to do with actually introducing some of these neurofibers and so on, but that's been difficult. So, as far as actually being able to get them in a dish or in an animal model and try, obviously, both the therapeutic effects as well as, proper lineage tracing for plasticity testing, so far, we haven't actually succeeded.
Yeah, okay, I understand. So it would be really nice if you can put them in culture and be able to propagate them and to work with. So thank you again, Peter. So we will move forward and see you soon. So now we will move to the third speaker, so Mariella Philbin. So she is co-director for Research Pediatric Neuro-Oncology Program, assistant professor, Harvard Medical School, and associate member at the Broad Institute of Harvard and MIT. So Mariella is MD PhD. She joined the faculty at Dana-Farber Boston Children's as a pediatric neuro-oncologist in 2017. So she's, of course, as MD in charge of patients with brain tumors, and she also focuses on establishing cellular networks of tumor dependency in pediatric brain tumors with the goal to find new treatments. So she made significant advances using single-cell transcriptomics and specifically in pediatric brain tumors. So today she will present a talk on this topic. So we will move to the last speaker of the session. So the last speaker is Jill MESIROV. So she is Associate Vice Chancellor for Computational Health Science, Professor in the Department of Medicine at UC at San Diego, so University of California. So Giles is a former mathematician, and then as computational scientist, she spent many years working on computing on problems that arises in science, engineering, etc. And then she moved to the biochemical research. Her research focuses on cancer genomics using machine learning, basically in order to stratify risk patients or to identify candidate compounds for new treatment. She made tremendous advances in the field of computational biology, among them GACA, so gene set enrichment analysis that almost all of us know and are using on a daily basis today. So welcome, Giles, for your presentation. So we are done with the first part, and now I will shift in French. So, je vais parler en français à partir de maintenant. Donc, je ne sais pas si Pierre-Yves est toujours avec nous. Donc, dans cette session qui nous reste maintenant, donc qu'on a préparée avec Patricia, avec Pierre-Yves, nous allons aborder justement la question. Ah, Pierre-Yves, je vous vois, tout va bien?
Voilà. Tout va bien.
Très bien. Donc, on a 20 minutes, 20-25 minutes pour justement débattre, discuter, poser des questions sur le sujet, donc comme on a dit au début, des big data, que sont les big data, à quoi ça peut servir, etc. Donc, on va commencer la discussion par une discussion entre nous. Et au fur et à mesure que les questions vont arriver sur le chat, je vais pouvoir les lire et on va pouvoir discuter autour de ces questions. Donc, n'hésitez pas à y continuer. Donc, c'était très bien depuis le début. Là, on avait énormément de questions. N'hésitez pas à continuer et à poser vos questions sur le chat. Pierre-Yves, vous avez peut-être quelque chose, un commentaire où vous voulez commencer?
Oui. Alors, d'abord, moi, je voulais dire que j'ai été très impressionné par les 4 présentations qu'on a eues et je voudrais vraiment remercier les intervenants parce que à titre personnel, ils m'ont beaucoup apporté et notamment, ils m'ont permis de toucher du doigt l'importance du traitement de la donnée dans le traitement des cancers et notamment ceux des enfants. Alors, on a beaucoup entendu parler de volume de données, on a compris qu'il y avait des tsunamis de données, on a aussi compris que dans certains cas, on en manquait, on a compris que ces données ont besoin d'être partagées. Alors, Olivier, moi j'ai envie de vous poser la question, peut-être une première question de base. Pour vous, le traitement de la donnée et peut-être plus largement les big data, c'est quoi?
Alors, c'est une excellente question. C'est vrai qu'on parle de big data depuis tout à l'heure. Le mot big data, déjà en anglais, si on essaie de le traduire en français, veut dire des volumes exceptionnels en termes de résultats, en termes d'informations. C'est un mot qui n'est pas réservé au domaine du cancer ou au domaine de la science. C'est un mot qui est utilisé aussi dans l'industrie, qui est dans la télécommunication. Et globalement, dans la biologie du cancer et dans ce qui nous intéresse aujourd'hui, les big data sont essentiellement des données à partir de la tumeur, c'est-à-dire des données de séquençage, associées à des données également, des données cliniques, par exemple, des données qui peuvent être recueillies au cours du parcours de soins du patient. Donc, l'ensemble de ces données constitue ce qu'on appelle les big data. Tout ça remonte, comme l'a expliqué le Dr Olivier Delattre hier, l'avènement, le début, on va dire, de l'arrivée des big data a été vraisemblablement le séquençage du génome humain, donc dans les années 90, et pour l'arrivée en 2004, qui a constitué un effort énorme, certainement plusieurs milliards d'investissements, à peu près 15 ans de travail, je crois. Et on a cru effectivement qu'à partir du séquençage humain, on allait pouvoir tout définir. Mais non, comme on l'a vu dans les présentations, maintenant, il y a donc l'ADN, donc le séquençage de l'ADN, il y a l'ARN, donc dans les années 90, on a commencé à séquencer. Ensuite, il y a eu donc les protéines dans les années 2000, et donc effectivement, l'ensemble de ces données constitue les big data. Aujourd'hui, un génome, c'est fait en quelques heures et ça coûte moins de 1 000 € par exemple. Donc ça avait répondu à votre question, Pierre-Yves?
Oui, tout à fait. Et alors, vous parliez tout à l'heure et on l'a entendu aussi dans les exposés, de gros volumes de données. En gros, c'est pour caler un peu les esprits, ça Ça représente quoi, ces volumes de données? À quoi vous êtes confrontés?
Oui, on a entendu Peter, effectivement, qui parlait, pour le single-cell RNA-seq, d'effectivement, il a commencé à parler qu'il fallait plus d'échantillons, plus de cellules, mais qu'en même temps, ça donnait des volumes extrêmement importants. Juste pour vous donner quelques informations, un génome, c'est environ 150 gigas. Sur un ordinateur moyen d'aujourd'hui, on est à peu près à 500 gigas, ce qui fait que si on a une cohorte à peu près de 300 patients, ça veut dire, pour uniquement le génome, on est à peu près à 100 ordinateurs. Donc dans la vraie vie, dans les laboratoires, ce n'est pas du tout ce qui se passe. On a donc des systèmes de clusters, de stockage, avec des ordinateurs hyper puissants qui nous permettent de stocker et donc de faire des calculs. Même chose en termes de calculs, il nous arrivait de lancer des calculs pour plusieurs semaines, par exemple. Donc effectivement, il y a eu un effort extrêmement considérable avec l'avènement de la technologie, un effort considérable de l'informatique et de la bioinformatique pour gérer ces informations.
Alors, là, on voit bien qu'on est face à des volumes de données qui sont absolument considérables, mais est-ce qu'il y a des différences, par exemple, entre le traitement des données des adultes, concernant les adultes, et ceux concernant les enfants?
Le traitement même des données, non, mais ce qui est important, et ça a été mentionné encore une fois, plusieurs fois aujourd'hui par Steven et par Giles, c'est la notion de la quantité de données. C'est-à-dire qu'on on parle de 2500 diagnostics par an pour les tumeurs pédiatriques en France. Maintenant, pour d'autres types, par exemple, où effectivement il y a des big data pour les cancers adultes, notamment les cancers du poumon, là on a des volumes effectivement. Je crois qu'en France, on est à peu près à 45 000 diagnostics par an. Donc on n'est vraiment pas dans la même dimension. Donc effectivement, comme on disait, comme plusieurs présentateurs ont présenté, c'est vrai que pour pouvoir réunir une cohorte suffisante, parce qu'en bioinformatique, l'intérêt et le besoin, c'est d'avoir une cohorte suffisante de patients pour pouvoir, d'un point de vue statistique, pouvoir tirer des conclusions. Donc ça, c'est extrêmement important. C'est une différence. Véritablement, c'est une spécificité au niveau de la pédiatrie, comme on l'a vu hier aussi. Parmi ces cancers, on peut les diviser en 100 différents types de cancers.
Alors, on parlait de différents types de big data tout à l'heure, vous en avez cité quelques-uns. Est-ce que, selon vous, il y en a d'autres qui vont arriver prochainement? Est-ce que vous voyez le champ de la donnée s'élargir dans les années qui viennent?
Alors, volontairement, je suis resté très simple tout à l'heure quand je vous ai annoncé le génome, le transcriptome, le protéome. Mais aujourd'hui, on est capable, il y a l'épigénome, donc les modifications épigénétiques qui peuvent réguler les gènes. Au niveau des ARN, on peut aller voir spécifiquement des types d'ARN. Au niveau des protéines, les protéines sont modifiées. Donc là, on arrive effectivement à un domaine énorme de possibilités. Quelque chose qui était très important et qui a extrêmement été bien présenté par Peter et Mariella aujourd'hui, c'est l'étude à l'échelle unique. On a bien entendu énormément de résultats depuis 10 ans, 15 ans sur la tumeur dans son ensemble, c'est-à-dire en considérant ou en essayant de considérer que effectivement la tumeur est un seul volume. Mais non, à l'intérieur de cette tumeur, les cellules sont différentes et le single cell RNA-seq, donc le séquençage à l'échelle unique, notamment avec le transcriptome, cherche à aller regarder chacune de ces cellules. Et là, véritablement, on a un point d'entrée extrêmement important pour essayer de mieux comprendre ce qui se passe pour ces tumeurs. Et Mariella, par exemple, c'est comme ça qu'elle a pu éventuellement trouver une cible et potentiellement un inhibiteur avec CDK6, par exemple, dans sa présentation. Donc non, ça va effectivement encore grandir.
Alors, les parents, enfin les patients et puis les familles de patients et les parents aussi se posent souvent la question de savoir ces données, en fait, à quoi elles servent, à quoi elles vont servir. Alors, on a eu quelques exemples, enfin, on a eu 4 très belles études présentées tout à l'heure, mais plus largement, à quoi elles servent ces données et à quoi servent les traitements?
D'accord. Donc, on me dit qu'il y a 2 personnes, donc il y a Giles et Steven, qui veulent intervenir et qui veulent parler pendant notre discussion. Donc, je donne la parole à...
Yeah. I just want to say, Olivier, you forgot metabolomic data. So we're just beginning to be able to acquire metabolomic data. and to actually be able to identify the metabolites that we're acquiring. So that's going to bring a whole new view of what's going on in these tumors. And, you know, the other thing that, the thing in some sense that drives the acceleration of the big data acquisition is the extent to which the cost of acquiring that data goes down. So we don't have as much proteomic data or metabolomic data at the moment because it's more expensive to get it. But technology is always improving in that regard. And so, you know, I think that will certainly drive it. But I would not be surprised if there weren't new kinds of data types. But I think metabolomic data is the only one that I think deserves mention that wasn't.
I would like to add, I think a lot more needs to be thought about in terms of big data. We've talked about everything that's measured once someone has cancer. If we're going to look at survivorship issues, we have to more effectively capture all of the treatment and the events that take place during the treatment of that individual. That's a lot of data to start to capture, from radiographic to radionuclide studies to chemistries to measurements. That is a very intensive set that we don't do very well, so that we can map and try and work backwards. So for instance, for acute lymphoblastic leukemia, the hard question is, can we treat some subsets of children with less therapy, meaning they have less toxicity, and still have the same outcome? Those are very difficult studies to conduct. We need that data, and there's a lot of big data for the clinical. And then I want to go a step further back. The ability to put together the risk factors, whether they're congenital anomalies, whether they're environmental things, looking at climate change and its potential effect on the environment, and what that may mean for diseases like cancer or affecting pregnancies, you know, because one of the things we really haven't talked about, a key issue in pediatric cancer is what's happened to the mother. And what can we categorize? What can we capture? What can we study in the placenta and the exposures? And for that, we do very, very little data. And there is a tremendous opportunity to collect an awful lot of information to understand how and why we get to where we get when we have that diagnosis. And, you know, as we saw in the Chernobyl study, there we had this very unfortunate accident, but we were able to see in a discrete environmental exposure. Usually it's not going to be so discrete. And I think that in order to pull out and understand what really happens, as opposed to anecdotally pulling things out, we need very large data sets. So I would submit there's going to be even larger data sets around all the genomic, proteomic, epigenetic characterization, which we have the technologies. Believe me, I'm a strong believer in that. I do that for a good part of my life, but I also do the other things, recognizing at some point they have to be integrated if we really are going to change the nature of pediatric cancer and think about prevention of pediatric cancer as opposed to dealing with it once it occurs.
So I would just make a friendly amendment to what you said, Steve.
Sure, go ahead, Jill.
I agree with you 100%. And I mean, clinical, that kind of clinical and other ancillary data is often the most difficult data for us to have or get access to in our studies. But as you gather all of this data, this wonderful data that you're talking about, I think the problem of privacy and protecting the patients to whom that data belongs,
no question,
even more significant. And, you know, we can't keep people out of our gas pipelines and our meatpacking plants. How are we going to keep people out of this very, very critical data, which can have a huge impact if it is breached, if it becomes available, right?
No question. I would counter and say that if you look at the studies of compliance of families of children with cancer who are asked to participate in studies or provide information or biospecimens, the success rate or the acceptance is phenomenally high. I would say most parents want to know why and what to do. And privacy issues become secondary there in that setting. I'm not saying that we leave them alone. I think they're tough things that we have to solve, but we can't say that we can't solve them. That's not acceptable.
No, I'm not saying that we can't solve them.
No.
And I agree with you that, you know, I agree with you that people do want to donate. They want, you know, their tumor tissue to be available for research. They want their data available. Absolutely, they do. That's been my experience also. But I think it's also incumbent upon us as the data guardians, so to speak, to pay very, very careful attention to how that data is held and how it's used and by whom it's used. That's the only amendment I want to make to what you're saying.
I completely agree. Vigorous agreement with you, Jill.
Well, we're done then.
Mais je vous remercie pour vos interventions parce que je pense que c'était un petit peu en lien avec la question de Pierre-Yves.
Oui. En fait, on est en plein dans les questions que se posent les parents et les familles, en fait, sur la confidentialité des données. Et on comprend bien la nécessité du partage de ces informations. Alors, il y a le recueil du consentement dont on sait qu'il doit être effectué. Olivier, ça vous pourrez peut-être nous en parler, mais il y a aussi les procédures qui doivent être mises en place pour le partage des informations. Comment on s'assure qu'effectivement les données sont partagées sont correctement partagées et en respectant la sécurisation de ces données. Olivier, comment est-ce qu'on peut le traduire ça?
Oui, alors avant de répondre à ces questions, je pense qu'il y a un point qui était important, qui a été soulevé justement dans la discussion, c'était la question de à quoi ça sert. Et ça, je pense que c'est extrêmement important. À quoi ça sert? Les big data, ça peut paraître abstrait, ça peut paraître effectivement difficile de comprendre à quoi ça peut servir. Mais de façon concrète, des big data peuvent servir par exemple effectivement à une meilleure caractérisation et surtout une meilleure caractérisation et un meilleur diagnostic de ces tumeurs qui sont extrêmement complexes. On est à peut-être une centaine de différents types de tumeurs. Et effectivement, il existe des méthodes aujourd'hui pour justement pouvoir, notamment le méthylome par exemple, être capable de savoir pour un meilleur diagnostic. Ensuite, ces data, comme on l'a vu pour CDK6, peut-être pour CD7 par exemple, ça peut mettre en lumière des nouveaux traitements. Et ça, c'est aussi assez important d'essayer de pouvoir, grâce à ces big data, de pouvoir trouver de nouvelles thérapies ciblées. Pour revenir à votre question, Pierre-Yves, un échantillon qui est séquencé, il y a bien entendu un consentement famille, consentement patient, non seulement consentement pour la recherche, mais aussi pour l'informatisation des données, deux choses qui sont essentielles. Ensuite, immédiatement, effectivement, ces données sont ce qu'on appelle anonymisées, c'est-à-dire que il y a un code qui est donné à une séquence, par exemple, et dans le cadre, par exemple, de partage, lorsque l'on publie, un exemple simple, ce que Jal a présenté et ce qu'on a co-publié ensemble, effectivement, les résultats qu'on a obtenus, on les a mis en ce qu'on appelle en anglais open access, c'est-à-dire que les résultats, les séquençages, etc., les données sont accessibles. Ça veut dire quoi? Ça veut dire que vous pouvez les utiliser. Donc ça, c'est voilà. Et dans l'esprit de ce partage, les données sont mises sur notamment ICGC ou EAG, ce qui sont donc des serveurs où vous pouvez uploader, donc ajouter vos résultats, et ensuite il y a tout un système qui permet de pouvoir sécuriser l'ensemble de ces données. Nous-mêmes, dans les laboratoires, on a des systèmes sécurisés de gestion de ces données, du stockage de ces données, qui a un certain coût d'ailleurs, mais voilà, ces données sont sécurisées sur ce type de serveur.
Donc on comprend que les données et les modes d'accès sont effectivement sécurisés. En pratique aujourd'hui, Olivier, c'est quoi les challenges qui vous attendent pour faire parler ces données? Comment vous les voyez?
Il y en a réellement, je pense qu'il y en a un certain nombre. Néanmoins, je voudrais dire quand même, si on considère au niveau des big data, si on considère que c'est un véritable marathon, ça a été un marathon, je pense qu'on est en train de franchir le fameux mur, je dirais, du 35e kilomètre. Et tout ça grâce, effectivement, à des efforts énormes technologiques pour obtenir toutes ces données, des efforts informatiques. Ce qui est important aussi, c'est effectivement notamment les personnes qui vont travailler avec ces données. C'est extrêmement difficile de pouvoir effectivement fidéliser ou disons avoir des personnes qui vont pouvoir… On est vraiment dans une multidisciplinarité ici, donc beaucoup viennent du milieu de mathématiques, physique, et c'est extrêmement difficile. Donc on est véritablement en compétition, par exemple, avec des compagnies très connues qui ne travaillent pas du tout effectivement sur la biologie. Donc effectivement, des gens extrêmement compétents dans ce domaine-là sont quand même extrêmement importants. Maintenant, effectivement, le partage, la création de ces grandes cohortes est en train de se mettre en place. Et effectivement, je pense que depuis 5, 6, 7 ans, on a vraiment une accélération de la publication de ces données et avec un partage qui est totalement open access dans la plupart des cas.
Et peut-être je dirais une notion qu'on a finalement assez peu évoquée tout à l'heure, à moins qu'il y ait des questions dans le public.
Je regarde en ce moment, Pierre-Yves. Non, vous pouvez y aller, je suis en train de les regarder.
Alors, en fait, ça a trait à l'intelligence artificielle et donc qui est une voie supplémentaire d'exploration avec de temps en temps des questions qui peuvent être posées dans les traitements médicaux et dans la façon dont on va soigner les personnes. Est-ce qu'un jour, on peut être confronté à une intelligence artificielle qui déciderait d'un traitement avec l'avancée du traitement des données, avec la capacité qui s'améliore d'année en année à la fois en termes de calcul et en termes de partage d'outils? Est-ce qu'on peut être confronté à ces questions-là qui vont soulever des questions éthiques très claires? Est-ce que le traitement qui va être donné à mon enfant va être, ou à moi-même d'ailleurs, va être choisi par une intelligence artificielle ou par un médecin?
Alors c'est une excellente question et d'ailleurs c'est une question qui est vraiment beaucoup revenue dans le questionnaire qu'on a récolté par l'intermédiaire de l'UNAPEC. D'ailleurs, on remercie justement ce questionnaire qui nous a vraiment aidé à préparer cette session aujourd'hui. Alors, effectivement, je pense que le mot intelligence artificielle peut, pour le grand public, un petit peu faire peur. Effectivement, dans le début de l'intelligence artificielle, alors je ne suis pas un expert, mais je pense que dans les années 50, il y avait effectivement l'envie de remplacer, c'est-à-dire de remplacer par une machine les activités ou l'activité humaine. Je pense, et encore une fois, je ne suis pas un expert, je pense qu'on est un petit peu revenu par rapport à cette idée. Et on est vraiment dans ce qu'on appelle, d'ailleurs, Richard Gilbertson l'a très bien présenté hier, on est vraiment dans le deep learning, c'est-à-dire dans l'apprentissage. C'est-à-dire que la machine va pouvoir voir énormément d'informations. Par exemple, des images. On a des exemples, par exemple, de pouvoir aujourd'hui, grâce à cette forme d'intelligence artificielle, de pouvoir reconnaître, par exemple sur le cancer de la peau, de pouvoir reconnaître spécifiquement une image. Maintenant, la machine, l'ordinateur, a eu comme information, non pas 2, 3 images, mais 30 000, 50 000 ou 100 000 images associées avec des informations. C'est-à-dire, cette image correspond à telle pathologie. Cette image va correspondre à un pronostic bénin. Cette image va correspondre à un pronostic malin. Etc., etc. Énormément d'informations. Et donc l'ordinateur va, grâce à des algorithmes, essayer de définir un portrait, et c'est-à-dire de dire, voilà, on pourrait penser qu'effectivement, il s'agit de cette pathologie, ce traitement pourrait être. Et quand j'utilise le temps « pourrait », c'est vraiment de l'aide. C'est-à-dire que c'est un soutien à la médecine, c'est un soutien à la recherche, mais en aucun cas, je ne pense pas aujourd'hui que l'intelligence artificielle va décider et le médecin pourra utiliser ces informations dans le parcours global du patient. Je ne sais pas si ça répond à cette possible inquiétude de l'intelligence artificielle.
Ici, je pense que c'est une réponse qui est d'ailleurs de nature calmer certaines inquiétudes. On comprend bien, il y aura toujours quelqu'un pour décider, pour trancher et choisir.
Donc je regarde dans le chat. Je crois que nous n'avons pas d'autres questions. Je regarde le timing. Je pense qu'on est effectivement dans les temps, ce qui était quand même important. On avait quand même une longue session. Il est 19h20. Donc je vais conclure cette troisième session et donc cette journée. Donc tout d'abord, je souhaitais vraiment remercier donc les speakers et les participants, toutes les questions qui ont été posées aujourd'hui dans le chat. Je remercie tous les organisateurs qui ont vraiment beaucoup aidé pour la préparation de cette journée.